Hello and welcome to the Light-Sheet Chronicles. A decade ago, in 2014, Light-Sheet Fluorescence Microscopy, also called Single-Plane Illumination Microscopy or SPIM, was named the Nature Method of the Year for its ability to gently image biological samples in 3D, capture cellular and subcellular dynamics, and do so over long periods of time. Now in 2025, we take stock and talk with world-leading scientists and experts in light-sheet microscopy about the technology behind the microscopes, imaging across scales, how we can use lasers to manipulate tissues, biomedical image analysis for light-sheet data, and much, much more. I'm Dr. Elisabeth Kugler, a science communicator and your host of this episode, Introduction to Light-Sheet Microscopy, where we explore the technology behind the microscopes. Our guests today are Dr. Johanna Perens, specialist in 3D imaging and image analysis, and Dr. Hecksher-Sørensen, Director of Scientific Sales and Imaging Innovations from Gubra, a company which specializes in preclinical drug development. Welcome to you both.
Hello. Thank you.
So it's fantastic to have you both here today. And maybe, Jakob, you could start by introducing our listeners to Gubra and what you're doing.
Absolutely. So Gubra is a small biotech company in Denmark, and we have kind of a dual business, one where we work as a biotech company and one where we work as a CRO business for large pharma and biotech. And as part of this work in the service business, we use light-sheet microscopy to image the efficacy of drugs on either pathologies or other things you can think of, like various endpoints we can quantify.
That's amazing. Thank you. And with us today as well is from Bruker Luxembourg, the Light-Sheet division of Bruker, Dr. Malte Wachsmuth. He's the managing director of the Luxembourg GmbH. And Luxembourg is at the very heart of light-sheet microscopy development. So Malte, firstly, welcome. And maybe you could introduce Bruker Luxembourg to our listeners.
Yeah, thank you. Sure. So we are the Light-Sheet Microscopy division of Bruker, like you already said. We started as a spin-out from EMBL in Heidelberg, Germany, and then became part of Bruker rather quickly to become part of the fluorescence microscopy division. The other products in the fluorescence microscopy portfolio are 2-photon microscopes and super resolution microscopes, but we are entirely focused on light sheet microscopy, complemented by big data solutions and also a screening solution.
Which is quite a wide portfolio, and I'm really excited. So we kind of really can dig in the next few episodes about everything there is about light sheet microscopy and data. So I'm really excited. And so maybe, Malte, also we can start with the first question by asking you to give us a brief overview about what light sheet microscopy is and how it differs from imaging methods such as confocal microscopy or 2P.
Sure. I mean, there's a lot we could talk about, but I will try to focus a bit now on light sheet microscopy. This is a method that has been effectively conceived in 1903 by two researchers, Zsigmondy and Siedentopf. They built the first, what one would today call a light sheet microscope. However, this then became quiet for like a century until the early 1990s. Where a first implementation of light sheet microscopy was conceived and published in the context of biomedical research. Then in 2004, I would say seminal publication from researchers at EMBL in the group of Ernst Stelzer in Heidelberg came out on imaging dynamics in zebrafish development using a light sheet microscope. So what is a light sheet microscope? When you think of a regular microscope, you typically have an objective lens that you use to basically look at an object. And when you illuminate it with laser light or with just a spectrum from a lamp, then you use the same objective lens typically. However, if you want to image a larger object, like a small embryo of zebrafish or Drosophila in 3 dimensions with high resolution, what is really a very useful addition is to illuminate your sample with literally a thin sheet of light. This is also where the name comes from the side, perpendicular to the optical axis of the objective lens I mentioned already. And then you use that objective lens just to take pictures of every plane that you're illuminating one after the other. This allows to really very gently acquire 3D data from small objects. And the big advantage is that it is very fast compared to other methods like confocal microscopy or 2P microscopy. and it is very gentle to the sample so that you can image over a very long time. Compared to confocal microscopy, what you do in a confocal microscope is you're basically illuminating one spot after the other, pixel by pixel, line by line, and then plane by plane of your 3D object, and you sequentially acquire information from your little object, whereas in light sheet microscopy, you do this in a fully parallelized way so that you're much faster. The 2P is, in a way, an alternative method to confocal microscopy. You do the same. You're illuminating a spot with an infrared laser, for example, and all the light emitted from this tiny illuminated spot is collected. And due to the fact that your illumination spot is highly confined, you know where all the light comes from. This is a method that can allow to go quite deep into tissue, for example, of a living animal brain, or into larger objects that are as big as, let's say, a millimeter or a few millimeters. But it's also a very sequential method, and therefore the big plus of light sheet microscopy is really that it's much faster compared to the other methods, and very strong when looking at live samples where you have to be fast to catch videos of rapid events, or very large samples where you just would want to save time, because if you acquire data and it takes on a confocal microscope a couple of days or even a week, then you're usually quite happy when you can save a lot of time and do this in an hour instead of a day. Which probably takes us also to the use of light sheet microscopy with you guys at Gubra.
Absolutely, because we love saving time, don't we? And getting lots of amazing data. And so, Jacob, that also really nicely bridges me over to you. And as Malte said, you at Gubra use cleared samples and tissue clearing to image, for example, large brains. And so it would be great to hear about the typical application that you use at Gubra, because I know you use a LCS-SPIM, which is a light sheet microscope that is designed for large cleared samples. So maybe you can tell us what you're using it for and what's your typical application.
So I think that just to give a little bit of background, it's a rapidly evolving technology, and over many years there's been different approaches that has been slowly developed. So one of the really important requirements for doing light sheet imaging is the clearing and labeling of the samples. So in many ways, you can imagine that using antibodies to label is very similar to what you do in histology when you look at different markers within tissue on sections. But there's also another way that you can actually label the tissue, which is injection of fluorescently labeled peptides into the animals while they're still alive, and then take out the organs. And once they've been cleared, you can then image the distribution of the molecule within the tissue. And this is actually how we started using light sheet, because at the beginning, the antibody labeling technologies were actually not good enough to label adult organs. So we were kind of stuck with imaging the biodistribution, which was also very nice for some applications. So for many people, many years, people have used radioactive tracers, as you also do today in PET, to look at the distribution of biological molecules. But in combination with SPIM, you can actually go in and you can get single cell resolution within your tissue of where your compound is actually going. So you can actually see at very, very high resolution the exact cells that your drug is targeting. And this was kind of the initial approach we took when I joined Gubra 7 years ago. We have then been expanding our portfolio of technologies to also include a lot of disease markers. So one example could be we do a lot of CNS imaging, where we look at how neurons are being activated in response to certain drugs. And as a company, we work a lot on metabolic diseases. So some of the things we're working a lot on is obesity drugs. And we look at how these drugs actually go into the brain and how they regulate appetite, or how they affect nausea, disease, or you can say side effects for certain drugs. By staining the brain with antibodies against certain markers, this gives us a very good idea of where in the brain the drug are going and how they affect the neurons within the brain.
That's amazing. Thank you. So you can use like transgenics, tracers, or antibodies to visualize different structures in the brain, and you use it to study drug distribution, and also to kind of work in drug development, but also use it in disease analysis. And you mentioned actually obesity drugs. And just as a side note, because it's in the news all over, have you been working with like recent obesity drugs such as Ozempic to study how they affect the brain?
Absolutely. So previous to joining Gubra, I was actually working at Novo Nordisk, and one of the key goals we had was actually to understand how Ozempic would get into the brain. I think that at the time when we started all this work, people didn't really know. We knew that people were losing weight when they got GLP-1 drugs, but we didn't know exactly how it happened. So we used whole brain imaging and biodistribution to actually understand how the compounds would enter the brain and regulate appetite.
That is super cool, because it really showcases that science isn't happening behind closed lab doors. It's actually a real world application, bench to bedside translation. And also that brings us to technological advances, because light-sheet microscopy has really various different geometries, which makes it kind of, difficult to talk about all of them. And we unfortunately don't have time to talk about all of them. But the setups have really developed and still are very much evolving. And because we can't really go into too much detail about all the different geometries, but Malte, maybe you can give us a little bit about like an overview about general geometries, why they're important, and how we can use them for different applications.
Maybe we can start with the geometry that you at Gubra, you guys are using. This is from our model, the LCS SPIM, LCS standing for Large Cleared Sample SPIM. And this indicates already that we develop our different products around the sample, basically. A major criterion at the beginning of a project, of a development project for us, is how can we actually mount a sample so that it can be imaged the best way without affecting the biology, without affecting the actual science you want to look at. And with the LCS BIM, the way it works is that you can mount your samples in a glass cuvette. So it's a glass container of, let's say, 20 by 20 by 20 millimeters with very, very transparent glass windows to the left, to the right, and to the bottom. The objective lens that I already mentioned, that is used for imaging, for looking at the sample, looks through the bottom part of the cuvette, and you have one light sheet entering this cuvette from the left side and another one entering the cuvette from the right side. They illuminate the same focal plane, and then the sample is moved plane by plane through these two light sheets, and the fluorescence signal acquired from the sample is then recorded on a very sensitive camera, so that you can acquire complete 3D stacks from, for example, a cleared brain that has been mounted inside this glass cuvette. The major advantage is that the objective lenses do not have to dip into the imaging medium, which is potentially chemically a little bit aggressive, and also swapping cuvettes allows for rapid changing of samples, so you can really optimize the throughput. If you want to image at live samples, another example from our product portfolio is the movie SPIM, standing for multi-view SPIM. The movie SPIM consists of 4 objective lenses that all look at a horizontal axis into a chamber. These are all so-called dipping lenses. That means they really dip and look into the mounting medium that you use to actually keep your sample happy. This puts quite some constraints on how you can actually put a sample between these 4 objective lenses. I will come to this maybe in 2 seconds. What you use 2 of these objective lenses are used for illuminating the sample, generating a light sheet in a horizontal way, and the other 2 objective lenses are used to image, acquiring a left view and a right view that are then to be registered and fused after the image acquisition. And one way to mount a sample, for example, a small developing Drosophila embryo, is to put it in a small rod of agarose and then hang it or support it from below to put it between these objective lenses, and then we can rotate it and move in 3D and look at it with high speed and observe it basically while it's developing and growing into, at the end of the day, an adult animal. And then there are more different more kinds of flavors of light sheet microscopes that we have, and they are all designed around how you can optimally mount your sample and how you can optimally observe your sample.
It's amazing to see there's so many geometries and applications. I myself always loved in vivo applications, and I think at the moment there's over like 130 different light sheet acronyms, and the list is growing. So there's definitely still much more development happening, which is amazing. And now I want to come back to Johanna, so Dr. Johanna Perens, and I'm extra keen to learn a little bit more about your work, because we know that light sheet microscopy produces large and often very complex data. And so I think that can be something that can be quite scary for someone who has never worked on a light sheet microscope. But in general, what is like your kind of insights into, working with light sheet microscopy data, and in terms of data structure and analysis, how to best work with them?
So like Malte already mentioned before, light sheet microscopy is a really unique way how it captures the data. So instead of single 2D images, we're dealing with very large 3D volumes. And then depending on the object size or the magnification we use for imaging, the object might be captured in several smaller subvolumes that need to be then assembled to reconstruct the whole object. So these image formats, for example, that Bruker uses, they are H5 file formats, and these allow to actually access smaller subvolumes in them directly without the need to load in whole images into the memory. So this makes image analysis a little bit more comfortable. So what makes this technique to stand out is its ability to show us the spatial distribution of molecular signals in intact organs. So not only are we looking at where the signals are located, but we can actually also study the morphologies and how structures are connected in 3D. So this means that we're actually dealing with a lot of data. And extracting meaningful insights manually, it's not just feasible anymore. So it means that light sheet microscopy requires actually solid computational infrastructures and quite advanced programming tools to even begin to work with it. So at Gubra, we have built high throughput imaging pipelines and also automated our image analysis workflows to handle exactly this, so that we can process our image data sets in batches and also apply robust segmentation and classification algorithms.
It's so nice to hear that. As image analyst, my heart is just like, it's amazing. It's great, especially as you say, working with smaller data packages, but also automation, standardization of tools and steps like segmentation is just amazing. But also you mentioned obviously large data and that you work with, for example, batch processing and a lot of automation, but that still means that you still have to handle large amounts of data, which can be often like a bottleneck and a challenge. So I was wondering, how do you handle these large amount of data and, copying and archiving and things like that?
So that is absolutely true. Light-Sheet is very data heavy. And for example, one raw image volume can range anywhere from a few hundred gigabytes up to several terabytes, actually. So it's quite a lot to handle. So to manage this, we rely on fully automated pipelines that takes care of image post-processing and data transfer. So when we first started to design our server setup, we did pre-calculations to estimate how many samples we are able to scan per day and how much data does it actually generate. This helped us to then plan ahead and make sure we have the right server capacity available all times. We use automated scripts that are then triggered every hour to move data from scanners to post-processing computers and then further on to storage servers where we access the data when we do image analysis. So, and this data transfer pipeline, this needs to run nonstop, basically, to make space for new scans. So this is how we kind of keep the wheels turning all the time. And another important step is obviously optimizing the scanning and post-processing settings early on. We test different configurations to find the lowest resolution that still gives us reliable quantification for the signal we're interested in. That way we can keep file sizes manageable without compromising the quality of the data and the results. So when it comes to analysis, we actually don't load the entire image volumes into memory. Instead, we load just subvolumes, especially for memory-intensive computations, for example, when running AI models for segmentations. A big part of our work actually goes into optimizing how we use memory efficiently.
I love that. And especially like that you say it needs experimental design before, and that you spend time on quantifying, how much storage do you need, what's the lowest resolution that you need, how to automatically trigger data transfer, and all these things which are so, so important to just keep, like you say, the wheels just turning, and that there's always kind of free space and always something happening in the lab, which is obviously great if you want to have high data throughput, right? But also on kind of handling large amounts of data, I think Malte at Bruker, we have a special solution, which is the Hive. Can you maybe speak to that?
I can only support what Johanna just said, that it's very useful to think about the experiment before you start it. The approach of, let's see what we get and then we can take care of this later can be a little risky when you're talking about terabytes of data. So, but we are aware that light sheet microscopes generate a lot of data, and that not everybody has the infrastructure or the resources or capacities in-house to build up their own infrastructure. So we have an offering that's called the Aquifer Hive, that is actually a big data storage and processing solution that you can put next to your microscope or next to your microscopes. And if need be, the microscopes can directly stream data upon acquisition on the Hive, which contains of a very fast and powerful storage module with up to 1 petabyte of capacity, and it's directly connected to a quite powerful workstation that allows to process data in a way you would like to. It has multi-user capabilities and functionalities to manage users, work groups, projects. And that's something we understood very early in the development process of our products, that this poses a problem to many people, and we would really like to offer a solution for this.
Which is, like we heard now, really, really important, not just because the data are large, but also obviously because then you can keep the lab ticking and always things going, which is really, really nice. And I think especially maybe centralized data processing is also something that we might want to mention. I think it's really important as well. We've now spoke a lot about like technological advances, but I also want to bring up one example of what you guys actually do at Gubra. So Jacob, back to you. Because I know that you had a really nice recent review in Drug Discovery Today, and the title of that was a 3D Light-Sheet Fluorescence Microscopy in Preclinical and Clinical Drug Discovery. And in that review, you had a really nice example where you spoke about studying whole mouse brains and the distribution of alpha-synuclein, and how you use that distribution to understand neurodegeneration and dementias, where in some subsets of these diseases, we see alpha-synuclein being distributed differentially. So maybe taking that as an example, Jacob, you can talk with us a little bit more about when you work with a whole mouse brain, what are some sample preparations that you need to do in order to get the best sample possible?
I think the reason for writing this review is when you work with Light-Sheet, I think most people are familiar with the beautiful images and videos that you can get when you scan whole organs. But the ambition we always had at Gubra was actually to also be able to extract these quantitative endpoints out of the data. So not only having the visual representation, but also get numbers out that we could use to qualify whether a drug is working or not. And part of this review was actually to illustrate and maybe characterize different examples of how we do this and how we work with it at various times. So for the Parkinson study, obviously some of the markers that you would like to understand is the ones that have been characterized to be involved with the disease progression. And one of these is actually a little protein called alpha-synuclein. It's weird in the way that it's actually present in the brain most of the time. But when you seed a fibril, which you can think about a little bit like when you like put an ice crystal into something and it then starts a crystallization process, this is almost what happens. So we actually put this preformed fibrils that we make in the lab into the part of the brain where we know it will then trigger this chain reaction. And we can then follow how it spreads. Now, I make it sound like it goes very fast. It actually takes a very long time for these crystals to spread over the brain. So some of the studies take almost half a year, but we can then monitor over time by taking out brains at different stages. We can see how the disease is spreading across the brain. And one of the places that the crystals then go to is the places that make dopamine in the brain. And once that happens, the cells making dopamine will actually start to die. And this is what then causes the Parkinson-like phenotype. So you can say a normal study that we would set up is basically to trigger this Parkinson-like reaction in the brain, and then wait a couple of weeks, and then we would start the treatment. And then we would take out the brains, and we would go in and we would stain for some of these markers. And maybe I should mention also a big thank you to some of the other things that are going on at the moment is the fact that there's a lot of effort in trying to make antibodies that are suited for light sheet microscopy, because I think this is one of the big problems in optimizing the stainings and the visualization of different endpoints is the lack of good antibodies. I think many of the antibody companies, they are now starting also to generate fluorescently labeled primary antibodies, which makes the whole process a lot easier. But we can then use these antibodies to go in and label the cells that make dopamine in the brain. And then Johanna and her team can go in and start analyzing and quantifying how many cells has been lost. Or if we start treating the mice with a certain drug, we can also visualize whether or quantify whether the drug actually has an effect on maintaining the cells and preventing the disease.
Yeah, it's really interesting to hear, because I think especially antibody stainings is often still a challenge, especially if you look at larger organs, for example, a whole mouse brain to do the stainings. So maybe as a side question, can I ask you about when you do the experimental optimization, you obviously need to spend a lot of time, not just on the tissue clearing, but also making sure that all the staining is homogeneous throughout the brain. Could you maybe talk a little bit about how you approach that to see if the staining is really, quote unquote, good enough for your experiments?
Obviously, it depends a lot on having good antibodies to begin with. But then you can say the challenge that we've had for many years was you need to optimize both the primary antibody and then the secondary antibody. And that can take many iterations to come up with the best amount of antibody you would use for a particular endpoint. Now, the problem we had with the alpha-synuclein is that when the thing you are then trying to stain changes over time, so you don't know exactly how much you will have in the brain at a given time point, that actually introduces like a third a variable that you have to control for, which is super tricky. And here, I think some of the really big advancements at the moment that is happening is this ability to directly conjugate your primary antibodies. So you kind of eliminate this procedure of also optimizing the secondary antibodies. And it sounds pretty trivial, but actually we have found it make a huge difference to how clean our stainings are. And I think also it allows the possibility of combining way more antibodies, because up till now, most of the good antibodies are rapid. So it's difficult to combine two rapid antibodies in the same tissue. But now that you can buy directly conjugated antibodies, that's no longer such a big problem.
Which is amazing to hear that technological advances really make a change, because I think, you said, it sounds like a simple problem, but I think everyone who has ever done an antibody staining or sat, and developed their own protocol, it's not trivial. It's not easy. And I think experimental kind of design and sample preparation is probably the most important step, right? Because if that's not right, you can't get nice data, you can't get a nice analysis. So getting that right is probably the most difficult thing of all.
Yes.
So yes, absolutely.
And unfortunately, it takes such a long time when you work with large organs, because the whole staining procedure is a month. So every time it doesn't work, it's another month.
Very time intense. And that's the thing, it's not just time, it's also obviously money and energy, and doing the experiments is very intense. Absolutely, I get that. And I think it's also the same then when it comes to your work, Johanna, that actually once you get a nice sample and you get really nice data and you want to do a bit more in-depth analysis, when you then look at whole organ accumulation, what considerations do you need to do? Again, maybe probably also similar to looking first if the signal is distributed nicely, but then also how do you do the 3D whole organ analysis of these data?
Yes, I also want to mention that it's very, very important to get the stainings to work really well, because if the antibody stainings don't come out that well and there is a lot of false positives and noisy data, it's very, very difficult to actually optimize or even develop algorithms for detecting the signal. So we definitely rely on very good images and image quality. But yes, so I think the very first big question actually is what do we want to measure, and what do we want to compare across groups when we do whole organ analysis? Because there's so many possible metrics you can actually quantify. So, for example, signal counts, or mean or accumulated intensities, or what is the density of the cells, or the coverage of different signals, for example, in case of alpha-synuclein or plaques. You could also look at the network features or distances to certain structures. I mean, you name it. And that choice often determines everything else, from scanning parameters to training of AI models. So one big challenge is actually the variation in the data, because we do high throughput imaging at Gubra, meaning that we actually acquire, in one study, we might have 50 to 100 samples. So that's already a variation within the samples, but then also within these large 3D volumes, you also actually have a lot of variation. So the signal might vary massively between treatment groups and between disease models or healthy and diseased organs. And obviously there are also image artifacts. Some come from tissue clearing or mounting or the scan itself. So there's another layer of variability on top. So what it means actually is that traditional analysis tools usually struggle to deal with that kind of inconsistency. So we use AI models for segmenting our data more and more. And for training AI models on this type of data, we try to include as much variation as possible in our training patches. And what is really important is that we have gone the way that we would rather want to have a small number of very high quality annotations that are prepared manually for training our AI models, compared to, for example, having a lot of data that is not annotated very well to begin with. Yes, so in general, we hope that this approach would help to our AI models to generalize better.
That's amazing. So did you maybe also compare actually which one performs better, like a small batch of really high quality annotation or a large batch of maybe less quality, more varied annotations?
Yes, we have actually tried out different setups, so to say. And because sometimes you can also prepare data the way that you use classical models to segment signals, for example, and then maybe clean it up a little bit and then feed it into the AI model. But then you have, because if you use classical algorithms for first preparing some data to start with, you can produce a lot more data, obviously. And however, cleaning it up is not always that simple because you do it manually. And so we have noticed that when we have then, let's say, 10, 20 very, very good samples that are really well annotated, this actually beats the other approach totally. So yes, we have done a lot of comparisons, yes.
That's amazing. And maybe one last question on that, because I'm really curious. I could speak with you hours on that. But just as a side question, so if you say you had maybe like 10 to 15 really well annotated data, how long does it take to, on average, annotate one of these data sets?
Oh, that's a tricky question. Yeah. Especially because I think it depends of what signal type it is. So sometimes if you have, for example, nuclear stainings they're amazing. They are simple to annotate. And you can also use actually some programs that are available to kind of help you. So they are pixel classifiers. For example, there exists a program called Elastic, or even Fitchy has a plugin called LabKit where you can you draw a little bit, like few pixels, and or like you annotate let's say, the cell background and the tissue, and you actually already get quite a good prediction. And then there are more tricky signals. For example, I would say alpha-synuclein is actually more tricky. Or, so basically signals that have large intensity variation inside all these image volumes, they are trickier because then pixel classifiers are not really good at it. Yes, so how long it will then take might actually take, I think, sometimes half an hour to even make one patch. And then obviously we kind of QC them. We very often also do an AI model first with these patches, and then we notice that it's actually not segmenting some other areas, not good enough, and then we go back to the patches or add patches to the training set. So in total, it might actually take several weeks of annotations and several rounds of training the AI model again and again.
Yeah, that's really interesting because it's like from the experimental design to then the stainings and everything, it takes weeks and weeks to then also do the analysis. It's really interesting to just get a feel for the timelines, because obviously every sample in the experiment is then very precious as well. We know that there's several hot topics, and obviously I could talk with you guys for hours and hours, but we don't have that much time. So maybe we can just talk about one hot topic, which is organoids, and maybe Malte, you can share your perspective on them with us.
Yeah, we understand that many of our users are interested in using organoids and use organoids for imaging, and organoids are something like a, miniaturized or simplified version of an organ typically derived from stem cells, and then are treated, let me say, appropriately. That's my level of knowledge here for that, because this is a technology that has been developed for example, by Hans Klevers in Utrecht over the last 10-15 years. And basically the trick is that you develop a 3D tissue structure out of stem cells, or this can also be derived from patient biopsies, for example, that share as much as possible in terms of structure, physiological conditions, biochemical signaling pathways with a certain type of tissue, like an organ, for example, liver, for example, the brain. And the big advantage is that it's compared to animal organs taken from animals, it's reasonably easy to generate. It does not require this high regulatory and also financial effort to basically use animals or organs from animals for imaging. And you still have a lot of the properties of the organ you want to study. So therefore, organoids have really emerged or keep emerging as a very widely used tool in biomedical research and in drug research, for example. And organoids are fairly complex three-dimensional structures, typically in the size of 200, 400 or more micrometers in diameter. And imaging organoids is as challenging as imaging, for example, the whole mouse brain. And then also light-sheet microscopy comes into the game again and into the equation, and really allows to image these 3D structures with pretty good resolution, with high speed, and with very gentle and low phototoxicity, so that you can basically observe the development, the growth, and maybe also the disease properties that you would want to model with these organoids with high resolution and over a long time. That's one application for organoids, is really the imaging of living organoids over a couple of days, for example. Also clearing of organoids and basically performing endpoint studies is also a wide used approach here. And the big advantage is really that you do not have to go through animals. You basically are a step back. The ultimate experiment is probably still requires to involve actual organs from animals. But this is as close as you can get to that without having to go through all this. And with our products, for example, the TrueDive 3D, that's one of our products that is really optimized for organoid imaging, because it can take very delicate samples, hold them in an incubator for days, and you can image them with high resolution every 30 seconds, for example, for a couple of days, and get, again, a lot of data, but that are also extremely insightful if you want to understand how an organ develops, how an organ turns into an unhealthy state, for example. I think this has a lot of potential, and we see that this is really developing very dynamically, and more and more people are actually interested in using light sheet microscopy for imaging of organoid structures.
So it's super exciting. There's much, much more to come. But this is all that we have time for today. So thank you to our wonderful guests for your time and expert insight.
Thank you.
Thank you.
Thank you.
It was great talking to you all. And so today we have talked about how light sheet microscopy works in principle, what some applications are, and how whole organ imaging can be used for drug discovery. So to our listeners, if you want to know more about organoids, make sure to check out the recent Bruker ebook on 3D cell cultures and organoids, which we will be linking below. And so again, huge thanks for tuning in. It was really a pleasure to be hosting this first episode on the Light-Sheet Chronicles. And in our next episode, we will be talking about imaging across scales, where we will talk about light sheet microscopy from subcellular structures to whole organisms. So make sure to tune into this. Thank you. Bye.
This podcast was sponsored by Bruker GmbH, the light sheet division of Bruker's Fluorescence Microscopy division. Bruker is a manufacturer of scientific instruments for life sciences, materials research, and industrial or applied analysis. If you'd like to learn more, please visit www.bruker.com/light-sheet. This podcast was produced by Keep It Science.